So, um, last week I talked about the light reactions and how inside the thylakoid membrane you've got photosystems, which are groups of pigments, and they capture light energy and convert it to chemical energy in the form of two molecules. We have ATP. Oh, sorry. ATP and NADPH. These are the molecules that carry the energy from light into chemical energy for the Calvin cycle. So the second half of photosynthesis is the Calvin cycle. This is where we're going to produce organic compounds using the energy that is stored in those two compounds, ATP and NADPH. And remember what I said before about how in order to make ATP, you need ADP and a phosphate. So when we made ATP, we had to combine ADP and an inorganic phosphate. And these two things came from the Calvin cycle, and they go to the light reactions, and then the light reactions produce ATP, which goes back to the Calvin cycle, which then goes to make ADP and inorganic phosphate. Same thing with NADPH. We have NADP plus and a hydrogen that came from the Calvin cycle. They go to the light reactions. It combines with the electrons to form NADPH. Oops. NADPH, which then goes to the Calvin cycle. So all of these things are related. And that's what I want to make sure that we keep our focus on, the fact that everything that we use in the light reactions came from the Calvin cycle, and everything that's used in the Calvin cycle came from the light reactions. And that's why they're called a biochemical pathway. This cycle is named after Melvin Kelvin, who was the scientist who worked out the details of this pathway. It is going to be a series of chemical reactions. And I don't even really care so much in the grand scheme of things if you remember every single step of the chemical reaction, because when you take, or if you take Bio 101 in your first year of university, you will most likely do all of this again. You'll learn it all again. Okay, so the second set of reactions, this is going to occur in the stroma. So again, I want to emphasize the structure of the chloroplast. Put this down. Okay, so we've got our chloroplast here. We have our stacks of thylakoids, and then we've got like these bridges that connect them like that. So our light reactions occur, that green is probably not a good idea, green on green. Light reactions occur in the membrane of the thylakoid, and the Calvin cycle is going to occur in the stroma, the solution outside of the thylakoids, but we're still within the inside of the chloroplast. And that is why the structure of the chloroplast is so important, because you've got these inner membranes in the thylakoids. And even within the thylakoid, if you remember, if this is one thylakoid, you are splitting water, and you've got a buildup of hydrogen on the inside of the thylakoid. And then you've got this big enzyme called ATP synthase. And because there's a low amount of hydrogen outside, all of these hydrogen ions want to diffuse through ATP synthase. And that's what drives the chemical reactions to combine ADP and a phosphate together. So you've got like membranes within membranes within membranes, all of which you are using to create concentration gradients for different types of reactions. And ultimately, you're still inside the one organelle, which is the chloroplast. And that's why the structure is so important. The Calvin cycle produces organic compounds, which is the main point, okay? I want you to also remember at the back of your mind that the main point of photosynthesis is to make food. and that food is in the form of organic compounds. And it's going to make organic compounds using the energy that's stored in those two chemicals that were made during the light reactions. Okay, so basically what we're doing is we're going from light energy, we're converting that to chemical energy, and we're using that to make food in the form of organic compounds. The process that we use to make the organic compounds is called carbon fixation. And it's really important to understand what that means, because carbon dioxide is not an organic molecule. It is inorganic, but you're going to convert it into an organic compound in the Calvin cycle.
The Calvin cycle is a series of reactions that results in conversion of carbon dioxide into the organic molecules needed to build new cells. It occurs in the stroma, which is the area of the chloroplast surrounding the thylakoid membrane. During the Calvin cycle, carbon dioxide, CO2, is added to a 5-carbon molecule called RUBP. The resulting 6-carbon molecule is unstable and quickly splits into 2 3-carbon molecules called 3-phosphoglycerate. Using energy from ATP and reducing power from NADPH, which are products of the light reactions, the pair of 3-phosphoglycerates move through a series of reactions and are converted into 2 molecules of glyceraldehyde 3-phosphate. When several of these glyceraldehyde 3-phosphate molecules have been produced, some combine to make glucose, while others are reused in the Calvin cycle. To generate an entire new glucose molecule, the cycle has to turn several times, because each turn of the cycle adds only 1 carbon atom from each molecule of carbon dioxide.
Okay, so that's pretty much the whole cycle. And I know there's a lot of steps, so we're going to go through step by step and look at what happens at each point. So this is an overall view of the Calvin cycle. You can see that you have carbon dioxide that comes in. There's a reason why they say 3 here, and I'll talk about that a little bit later. But ultimately, again, the main point is that you want to make organic compounds. Okay, so first step is CO2 is going to diffuse into the stroma, and an enzyme is going to combine the carbon dioxide with a 5-carbon molecule here called ribulose biphosphate, or RUBP. So that's here, 1, 2, 3, 4, 5. Okay. So you've got 5 carbons and a phosphate at each end. That's what the P is. Now the result of that, which they don't include, but I'll put that over here, that's going to be P, and then 1, 2, 3, 4, 5, and I'm going to use blue for the representation of the carbon that comes from carbon dioxide. Okay. This is a 6-carbon molecule, but this 6-carbon molecule is very unstable. And so it immediately separates in half to form a molecule called 3-PGA. 3-PGA stands for 3-phosphoglycerate, but you can just remember 3-PGA. Abbreviations are always okay. Okay, so we've got CO2, which has 1 carbon in it, and you add it to a 5-carbon molecule, and you make this 6-carbon molecule. But again, that blue C is the one that came from CO2. Because this 6-carbon molecule is unstable, it splits in half, and you end up with 2 molecules of 3-PGA. Now, 3-PGA is going to be converted into a new 3-carbon compound called G3P, glyceraldehyde 3-phosphate. This is going to happen in 2 steps, which you see here and here. Again, just ignore the numbers in front of the molecules for now, okay? The numbers don't matter. So the first step is going to be that each 3-PGA is going to receive a phosphate from ATP. So if you remember what we started with, we have 1, 2, 3, 4, 5, whoops, that's supposed to be blue, 6 and a P, okay? That splits in half to form this, 1, 2, 3, 1, 2, and then that's blue, okay? Now you're going to use ATP, and you need 2 ATP. So you take a phosphate off of each one. So you're left with 2 ADP. And this goes back to the light reactions. Okay. These 2 phosphates are going to be put on the other side of your 3-PGA. So one of your 3-PGAs becomes PCCCP. That comes from this one. And then the other one is going to be PCC blue C P. Okay, that's step 1. Then the next step is going to involve reducing NADPH to NAD+. So in this case, I'm going to go to the left side of the screen now, okay? So I'll rewrite what we have. We have PCCCP, and we have another one, PCC with a blue C, P. Okay, now we have 2 NADPH. We're going to take off 2 hydrogens. Those 2 hydrogens are going to replace the phosphate that we just added. So you're going to end up with 2 NADP+, which go to the light reactions. So then these 2 become PCC. And just to make things easier, I guess I'll add the hydrogen, although we don't usually look at, don't really pay attention to hydrogen. And we still have that blue. Okay. And so now the resulting compounds here is called G3P. Everyone okay so far? Okay. Here's the problem. We need to fix the inorganic carbon in carbon dioxide. We need to make it organic. We can't do that if we just take 1 carbon dioxide at a time. So here's where I point to this one. You've got an organic compound, but it's made of 2 organic carbons and 1 inorganic carbon. You can't, that doesn't do anything, doesn't help. And so this is why, I don't know. Oh, what happened to the diagram? Wait a minute. Hold on, I need to go back to, there we go. Okay, this is why you see the number 3 here. Okay, remember at the end we had P, C, 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 and then I just put an H there, and then H, C, C, and 1 blue C. And this is what we want, right? This is G3P. But the problem is we still have that inorganic carbon. So this is why we need to have 3 carbon dioxides, so that in the end, we have this instead. This is what we want. We want all 3 carbon dioxides to be fixed into an organic molecule. Because in the end, if you multiply everything times 3, now I'll talk about why everything's times 3. So you want an organic molecule of G3P to be made from you want it to be made from carbon dioxide. Because it's a 3-carbon compound, that means you need 3 carbon dioxides to come in per turn. That means that those 3 carbon dioxides combine with 3 molecules of RUBP. And when those 3 molecules of RUBP split up, you're going to form 6 molecules of 3-PGA. Right? If you start with 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6. So if you've got 3 of these and they split in half, you're going to end up with 6 G3Ps, or sorry, 3 PGAs. But the difference is, for this one of these has been made from carbon dioxide because you used 3 carbon dioxides. So when those 6 are separated, that's why you need to use 6 ATP and you produce 6 ADP. So then you're going to have phosphate on each one. And then you're going to reduce 6 NADPH, releasing a hydrogen. They're going to release those 6 phosphates. And then guess what? You've also got phosphate here, which can go back to the light reactions. So what you're doing is you are recreating, right? So we used 6 ATP and 6 NADPH. Those are the 2 molecules that are storing the energy from the sunlight in chemical energy. And now we're going to use them, and we're going to release the ingredients that we need for the light reactions. So we can go back and make more ATP and more NADPH to come back here and do the same thing all over again. But in the end, the main idea, the main thing that I want you to remember is that of all of these 6 of all of the 6 3-carbon compounds, only one of them was created from the carbon dioxide. That one at the end is going to leave the Calvin cycle, and they will be combined into glucose, because glucose is a 6-carbon compound. So what you're going to be left with is 5 molecules of, oh, hold on, that just came in. So what you're going to be left with is 5 3-carbon compounds. So you're going to have this. That's 3, 4. I'll put the next one. Okay. And then your other one is the G3P that's going to be used to make organic compounds here. The other 5 are going to be rearranged and reused to reform 3 molecules of RUBP. So you've got 5 times 3-carbon compounds, and you have to convert them into 3 times 5-carbon compounds, which is here. So let's just go ahead and rearrange those, and you'll be able to see why we need an additional ATP here. Okay, so first I'm just going to start making my RUBP, which you can see here is a phosphate with 5 carbons and another phosphate at the other end. So I've used 1 phosphate and then carbon, 1, 2, 3, 4, 5, and another phosphate. I'm going to make another one, 2, 3, 4, 5. So I've used a phosphate and 5 more, 1, 2, 3, 4, 5, and another phosphate. And then the last one, P, which is here, the last 5 carbons, 1, 2, 3, 4, 5. But you can see that I'm short a phosphate. So I'm going to need to have ATP here. And again, because everything is times 3, that's why you need an additional 3 ATP, and you make 3 ADP to put that one at the end. If you cannot recreate RUBP, then the Calvin cycle stops, right? If you can't recreate the molecules that you started with, then you can't keep going. So it's important that we are able to regenerate RUBP in order to keep the Calvin cycle going. But out of every 3 carbon dioxide, you make 1 G3P, which is this guy. So each of these came from a CO2, and that's called carbon fixation. I do want to go back to connecting everything together. So in the Calvin cycle, you are producing ADP, you're producing NADP plus, and you're producing phosphate. And all of those are going to go back to the light reactions to be used in those reactions. So this is a really good diagram of explaining how they're both connected. And it's also a good explanation of linking it back to the overall equation of photosynthesis, which is carbon dioxide plus water yields, I'll use the same thing they used in the diagram, CH2O, which is sugar, and oxygen. Okay, so you can see that the water is required for the light reactions. You remember we needed to split water. Okay, Pang, that's fine.
Okay, bye, sure.
Bye. Never mind. Okay, so remember water is needed to replace the electrons that we lost in photosystem 2. So that's why we need water. We split water, and that's when we create the oxygen, so we know where we need it. That's where oxygen comes from. The hydrogens build up on the inside of the thylakoid and then go through ATP synthase that drive the reaction that way. And then we also know why we need CO2 here in the Calvin cycle, and also how it produces the sugar. But linking again between the 2 halves of photosynthesis, the Calvin cycle produces NADP plus, ADP, and phosphate. These 3 are used in the light reactions, right? They're combined together. So NADP plus combines with the electrons from water and the hydrogens to form NADPH here. And the other hydrogens that are here go through ATP synthase, and then that combines ADP and a phosphate to make ATP, which go to the Calvin cycle. We see how those are used, and then they again produce these products. So the 2 halves of photosynthesis keep the other half going. Oh. Okay. So of the 6 molecules of G3P that are created, only one of them came from the carbon dioxide. That is the only one that leaves the Calvin cycle to be used by the plant to make other organic compounds. The other 5 are going to be rearranged to make the new RUBP molecules. Okay, so just to go through quickly again, you've got 3 carbon dioxides that come in to the stroma. They combine with 3 molecules of a 5 carbon compound called RUBP, ribulose biphosphate. That's going to create a 6 carbon molecule. So it's going to look like this: 1, 2, 3, 4, 5, 6. But you've got 3 of them. So remember, it's times 3. That molecule is unstable. It's going to split in half and it's going to form 6 molecules of 3-PGA, phosphoglyceraldehyde, I believe. But remember, of these 6, 5 of them look like this, and 1 of them looks like this. 1, 2, 3. That's the one that came from the carbon dioxide. Okay, you're going to convert these 6 molecules into a different type of 3-carbon compound in 2 steps. The first step is you're going to use 6 ATP because we have 6 compounds here. You're going to take a phosphate off of ATP, which produces ADP. That goes back to the light reactions. And that means you've got 6 phosphates. You're going to put them on the ends here. Okay, that's step 1. Step 2 is you're going to take 6 NADPH. You're going to remove the hydrogen, which means you've produced NADP+, which guess what? Goes to the light reactions. You're going to take that hydrogen, and that hydrogen's going to replace the phosphate that you just put on, which means that you're releasing those 6 phosphates. And where do they go? The light reaction. So this new molecule that you have, again, you've got 5 of these, and you've got 1 that looks like this that came from carbon dioxide. This 1 molecule of G3P is going to leave the Calvin cycle to make organic compounds. The other 5, because you've got, remember, 5 times 3 carbons are going to be rearranged to form 3 times 5 carbon compounds. And for that, you need an additional set of ATP molecules to add that phosphate at the end. And boom, you've remade your 3 RUBP, so you can accept another 3 CO2.
You're facing a giant bowl of energy-packed carbon crunchies. 1 spoonful, 2, 3. Soon, you're powered up by the energy surge that comes from your meal. But how did that energy get into your bowl? Energy exists in the form of sugars made by the plant your cereal came from, like wheat or corn. As you can see, carbon is the chemical backbone, and plants get their fix of it in the form of carbon dioxide, CO2, from the air that we all breathe. But how does a plant's energy factory, housed in the stroma of the chloroplast, turn a 1-carbon gas like CO2 into a 6-carbon solid like glucose? If you're thinking photosynthesis, you're right. But photosynthesis is divided into 2 steps. The first, which stores energy from the sun in the form of adenosine triphosphate, or ATP. And the second, the Calvin cycle, that captures carbon and turns it into sugar. This second phase represents one of nature's most sustainable production lines. And so with that, welcome to the world's most minuscule factory. The starting materials? A mix of CO2 molecules from the air and pre-assembled molecules called ribulose bisphosphate, or RUBP, each containing 5 carbons. The initiator? An industrious enzyme named Rubisco that welds 1 carbon atom from a CO2 molecule with the RUBP chain to build an initial 6-carbon sequence. That rapidly splits into 2 shorter chains containing 3 carbons each and called phosphoglycerates, or PGAs for short. Enter ATP and another chemical called nicotinamide adenine dinucleotide phosphate, or just NADPH. ATP, working like a lubricant, delivers energy, while NADPH affixes 1 hydrogen to each of the PGA chains, changing them into molecules called glyceraldehyde 3-phosphates, or G3Ps. Glucose needs 6 carbons to form, made from 2 molecules of G3P, which incidentally have 6 carbons between them. So sugar has just been manufactured, right? Not quite. The Calvin cycle works like a sustainable production line, meaning that those original RUBPs that kicked things off at the start need to be recreated by reusing materials within the cycle now. But each RUBP needs 5 carbons, and manufacturing glucose takes a whole 6. Something doesn't add up. The answer lies in one phenomenal fact. While we've been focusing on this single production line, 5 others have been happening at the same time. With 6 conveyor belts moving in unison, there isn't just one carbon that gets soldered to one RUBB chain, but 6 carbons soldered to 6 RUBPs. That creates 12 G3P chains instead of just 2, meaning that altogether 36 carbons exist, the precise number needed to manufacture sugar and rebuild those RUBPs. Of the 12 G3Ps pooled together, 2 are siphoned off to form that energy-rich 6-carbon glucose chain, the one fueling you via your breakfast. Success! But back on the manufacturing line, the byproducts of this sugar production are swiftly assembled to recreate those 6 RUBPs. That requires 30 carbons, the exact number contained by the remaining 10 G3Ps. Now, a molecular mix and match occurs. 2 of the G3Ps are welded together, forming a 6-carbon sequence. By adding a third G3P, a 9-carbon chain is built. The first RUBP, made up of 5 carbons, is cast from this, leaving 4 carbons behind. But there's no wastage here. Those are soldered to a 4th G3P molecule, making a 7-carbon chain. Added to a 5th G3P molecule, a 10-carbon chain is created. Enough now to craft 2 more RUBPs. With 3 full RUBPs recreated from 5 of the 10 G3Ps, simply duplicating this process will renew the 6 RUBP chains needed to restart the cycle again. So the Calvin cycle generates the precise number of elements and processes required to keep this biochemical production line turning endlessly. And it's just one of the hundreds of cycles present in nature. Why so many? Because if biological production processes were linear, they wouldn't be nearly as efficient or successful at using energy to manufacture the materials that nature relies upon, like sugar. Cycles create vital feedback loops that repeatedly reuse and rebuild ingredients, crafting as much as possible out of the planet's available resources. Such as that sugar, built using raw sunlight and carbon, converted in plant factories to become the energy that powers you and keeps the cycles revolving in your own life.
Okay, so that's actually a better explanation because they're looking at it from an even bigger picture, which is making that glucose, which is a 6-carbon compound. So you can go back and rewatch that again. That's a really good example, because if you've got it going 6 times, you've got a total of 36 carbons. 6 of those are going to make the glucose, and then that means you've got 30 left over to go back and make 6 more RUBP. And then he shows you that very specific way that they go about doing it. So they're putting different ones together or whatever. You can go back and watch that later on. So what is the balance sheet? So in the textbook, they're only showing you how to create 1 G3P molecule, which of course, ultimately, I guess you would need 2. So you could technically, if you want to, just multiply everything in this chart by 2. So you could have, that would be 6, and this would be 6. That would be 12, 12, 12, 12, 12, 12, 12, 2. That would be 6 and 6. And then you would have, yeah, right? Yeah. So that would be enough to make 1 glucose. But if we're just making 1 G3P molecule and not really worrying about the glucose so much, how much ATP and NADPH are required to make 1 molecule of G3P, or sorry, 3GP, G3P, whatever. I don't care at this point. Because I'm like, I think it's G3P. I've just typed it wrong. So each turn of the cycle will fix 1 carbon dioxide molecule, but just like in the video, we talked about there's many versions of this cycle that are happening at the same time. So if G3, yeah, it's got to be G3P. Let me just change that because the other one's 3PGA. G3P, 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 yeah. All right. So you need 3 turns of the cycle to make 1 G3P molecule, and 3 turns gives you 1 G3P and 5 3-carbon compounds that you go to rearrange to form RUBP. So if each turn you know what? Never mind each turn, because that's just going to be whatever. Let's just go ahead and add them up. I've got 6 and ATP here plus 3, which is 9. So you need 9 ATP and 6 NADPH. That's for G3P. So if it requires, yeah, because it's right there. If 1 glucose is equal to 2 G3P molecules, then how much ATP and NADPH is used to make 1 glucose? Well, that would be this times 2. So 1 glucose would need to use 18 ATP and 12 NADPH. Now you might think, well, that sounds like you're spending a lot of energy to make 1 glucose, but in chapter 7, you're going to find out just how much ATP 1 glucose can release, and it's much more than 18. Do I have time? Yeah, I got 10 minutes. All right. So the last thing is alternative pathways. There are different ways that plants can fix carbon because of the environment that they live in. So the Calvin cycle is the most common form. And we call plants that use the Calvin cycle, we call them C3 plants because they produce 3 carbon compounds like G3P. But other plants are going to fix carbon in a different way and send them to the Calvin cycle later. These are going to be plants that live in hotter, more arid or dry climates. And the reason is because at the bottom of the leaf, if you look at, here's your leaf, that's the top of the leaf, right? If you look underneath it, well, you can't see it with your eyes because they're super small, but there's holes in the bottom of the leaf, which are called stomata. One, that's the plural, by the way. One singular is called a stoma, not to be confused with stroma, which is the solution inside the chloroplast. I don't know why they have to be named so similarly, probably because of the Greek or Latin that it comes from. But anyway, so if you think about it, CO2 is a gas, right? And H2O is a gas. So in the atmosphere, at least. So how is the plant going to get CO2 if it doesn't have lungs to breathe it in? And that's through the stomata. And so CO2 goes into the plant through the stomata. And we also have transpiration, which is the water that comes out through evaporation, also goes out through there. So if you live in a desert and your stomata are open during the day, when it's super hot, you might lose too much water through evaporation. And so that also means that the stomata are going to close to conserve the water loss. But if the stomata are closed, then it can't absorb carbon dioxide. And because the Calvin cycle and the light reactions are happening inside the leaf, that means that oxygen is going to increase because oxygen also leaves through the holes, and oxygen inhibits the Calvin cycle. So all of this basically means that plants that live in a hot, dry climate, they have to find an alternative way to do it because they can't have their stomata open all day during the day. So there's 2 alternate pathways. One is called the C4 pathway. Here you can see 1 stoma, and they're basically controlled by guard cells, which I'll talk about a little bit later, or maybe next year, I don't know. But basically they either swell up and bend like a kidney bean to open the hole, or they shrink and they close. But that's the hole that I'm talking about. Looks like a little mouth. So for C4 plants, they're going to fix carbon dioxide into 4 carbon compounds. So during the hottest part of the day, the stomata, they're going to be closed. An enzyme during this time is going to convert the carbon dioxide into 4 carbon compounds, because the process that does that can work even if there's a low concentration of CO2 and a high concentration of O2, where the Calvin cycle can't work. But this, the C4 pathway can. Later on, the 4 carbon compounds can go to the Calvin cycle and be converted into organic compounds. Examples of plants that use a C4 pathway are corn, sugarcane, and crabgrass. And the last alternative pathway is called the CAM pathway. These are plants that live in extremely dry environments. They're going to have their stomata closed completely during the day, and they're only going to open their stomata at night when there is no light. Now, that's why I don't like the description of the Calvin cycle as the dark reactions, because a Calvin cycle can still happen during the day. It just doesn't depend on light being present. So at night, the CAM plants are going to take carbon dioxide in and fix them into organic compounds. And then during the day, the carbon dioxide is going to be released from those compounds, and they'll do the Calvin cycle during the day. But the big problem is they can't have their stomata open during the day because they'll lose too much water. And so when they take it in at night, they'll fix it into different organic compounds and then just kind of reverse the process and release the CO2 during the day to form the Calvin cycle. So even during the day when the stomata are closed, these reactions are going to take these organic compounds and release the CO2 for the Calvin cycle. So it doesn't need to get it from the air. Examples of these kinds of plants are cactuses, who obviously live in the desert, and pineapple. So a quick summary, got 3 minutes. So photosynthesis happens in 2 stages. You have the light reactions where energy is absorbed from sunlight. That light energy is converted into chemical energy that is stored in 2 compounds, ATP and NADPH. Those 2 compounds carrying the chemical energy go to the Calvin cycle, the second stage, where carbon dioxide and that chemical energy is used to form organic compounds. Photosynthesis is an ongoing cycle. So the products of the light reactions are used in the Calvin cycle and vice versa. Otherwise, everything stops. But these processes are also used to make other organic compounds like amino acids, lipids, and carbohydrates. It's not just making glucose. Okay. Right, so this one is in your book, so you can look at that yourself a bit later, but this puts the whole thing together inside the chloroplast. So you have the light reactions where you use light energy, you're going to use water, and in return, you're going to produce oxygen. You're also going to produce NADPH and ATP, which you can see here going to the Calvin cycle, which is in the stroma. Sorry, I'm talking so fast, but I'm really trying not to go over time. During the Calvin cycle, you take in CO2, you use the energy from NADPH and ATP to produce organic compounds, and in exchange, you're going to also produce NADP plus ADP and phosphate, which go back to the light reactions. I'm out of breath. This, keeping in mind that you have thousands of chloroplasts inside of a plant cell, and you have gazillions of plant cells that make up a plant. So these processes are occurring at like an exponentially larger amount than just this one little thing. Okay, I think. Blah, 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 blah. Okay. Oh, okay, definitely going to save that for later because I am really out of time now.